So, an early theoretical, kind of approach is something known as the cell doctrine, right? Where basically all tissues, complex tissues in our bodies, for example, are, thought to be composed of smaller units that are known as cells. But when neuroanatomists started examining, the structure of the brain, when they made it through, the scalp and the skull and the meninges, the dura, the arachnoid and the pia mater, and they got into the gray matter and they got deeper, they would, cut, like coronally like this, or they would cut, horizontally like this, or sagittally like this. They would actually reveal, structures that didn't look like a typical cell. So if you think about what a cell looks like, what comes to mind when you initially think of that? When you think of just, a cell, an animal cell, well, most people think of something that's, rather spherical, right? Or perhaps kind of elongated. But neurons, and glial cells are actually rather remarkably diverse and unusual looking, assemblages of dendrites and axons and, collaterals and processes. They can look rather remarkable and rather uncell-like, at least to the early proponents of the cell theory. So there was a battle over whether, the cell doctrine actually applied to the brain. And whether there was a neuron doctrine, which is, misnamed now because there's obviously more than just neurons that are localized, in the brain. So Camillo Golgi was a very, very famous, neuroanatomist, an Italian neuroanatomist who developed a silver stain that permitted the visualization of individual neurons. He actually didn't realize, though, that these were separate cellular entities. He, in fact, proposed that the brain was a mass of interconnecting wires that was referred to as like a syncytium, is the sort of the Latin for this massive interconnecting, components. So he thought that, the wiring just sort of ran directly from one cell into another. While Santiago Ramón y Cajal, is an astonishingly famous also, neuroanatomist and also an artist who drew, what he saw under the microscope when he looked at segments of brain tissue, from the human brain. And his technique was actually to look at lots and lots and lots of Golgi-stained images of, cells from the brain, and then turn around and then draw, the sort of archetype of what he was observing. So his work is just utterly remarkable and beautiful and compelling. There's a Cajal Institute in Spain devoted to his drawings. Through Northwest Noggin, we've actually had the wonderful privilege, and honor to be invited to the National Institutes of Health in Bethesda, Maryland, where there is an art gallery, of local artists from the area who work with researchers and then, create works based on the work that's being done in those laboratories. But the centerpiece of this kind of exhibit is a Cajal exhibit, where, there are lots of, Cajal images of brain cells, basically, and brain circuits on display. So Cajal, believed that these neurons, he thought he saw gaps, that they were separate entities. And, the two of them apparently had quite a battle over this, and didn't like each other. And apparently they were co-awarded the Nobel Prize, actually in 1906, for the discovery of, the physiology of neurons, the structure of neurons. But Golgi didn't buy, the idea that that these neurons were separate entities at that stage, actually. And apparently there were almost fisticuffs on stage. What's interesting is that Golgi, to his credit, also identified other components of, the points of connection, between one neuron and the next, which is, of course, known as the synapse, right? And Golgi identified, the fact that there were all these little kind of extracellular kind of connections, it seemed, around individual synaptic, points of contact. Lots of little, what they call an extracellular matrix. These, it turns out, are composed of sugars and proteins that are extruded from, the membranes of both the presynaptic and the postsynaptic neuron. And this extracellular matrix or perineuronal net is actually, quite an important structural feature of neural networks, in that it helps to sort of, restrict further, development of particular synapses, or restrict the kind of development that is occurring around specific synapses. So while Cajal is famous for identifying neurons, as individual cellular entities, and doing this absolutely utterly remarkable artwork that displays these gorgeous cells, and I'll put some images on our site. Golgi also, even though he wasn't a huge neuron doctrine proponent initially, he actually also identified something called the perineuronal net, or the extracellular matrix that sort of forms around synaptic connections in our neural networks in the brain.